We're going to take a very fast world tour. We're going to visit Denver, Colorado, Durham, North Carolina, Kyoto, Japan, and bring it on home to wherever you live right now. And so I don't know how many of you can remember the first time that a particular event happened in your life. You probably do remember when someone introduced actives to you and who that person was. You may not remember when you first heard the term oxidative stress. I do, because I heard it from and was taught about it from a guy that knew a little bit about it. In fact, I not only heard it straight from the horse's mouth, so to speak, although I'm not sure he really likes being referred to as a horse, but I think he'll understand the phrasing. But it was from Dr. Joe McCord at the Webb Waring Institute for Oxidative Research in Denver, Colorado. And that was back in 2008. I had been contacted by a lawyer, actually, and the chairman of the board of LifeVantage to see if I would be interested in taking the helm of this small little company that had this product that I didn't know anything about. What's interesting is that I had heard about things like antioxidants and free radicals. I mean, I'd heard the phrases. I didn't really know what they meant, to tell you the truth. And at that point, I'd been in the nutritional supplement field for well over 20 years. But and when I first looked at the ingredients of this product called Protandim, I wasn't particularly impressed. I mean, I was familiar with all the ingredients and I thought, yeah, okay, not that big of a deal. I had not seen the ABC primetime video yet, which to this day remains the most positive piece ever aired on a supplement. And I had not, of course, had not talked to Dr. Joe McCord. So I flew out to Denver and was in his office at the Webb Waring Institute for Antioxidant Research. And at that point, he asked me a few questions because he is kind of professorial. And there are lots of things about me that drive Joe crazy. There's no doubt about it. But one thing he does like the fact that I'm very interested in science and I have a somewhat inquisitive mind. And so he said, well, do you know what an antioxidant is? I said, nope. He said, well, do you know what an oxidant is? I said, not really. He said, well, an oxidant comes from the word oxygen. It's also known as a free radical. And he said, let me tell you a story. He said, back when I was a grad student at Duke University in Durham, North Carolina, so now we're traveling from Denver over to North Carolina, he said, we were observing something. He said, back then, nobody knew what free radicals were. Some people had theorized about how they might exist, but probably not naturally. And he said, we noticed something in the tissues of various animals and plants. We noticed, I'm not sure if you call it a molecule, but they noticed something that was in everything that breathed oxygen. And he told about bringing in, how everyone would start bringing in animals. They'd bring in guinea pigs, they'd bring in rabbits, their dogs, their cats, they'd bring in pumpkins, they'd bring in spinach, they'd bring in all types of things. And in every single case, they found this stuff. And he said, you know, it was an answer in search of a problem. Well, that stuff was called superoxide dismutase. And of course, it had a role. It wasn't there by accident. In fact, it is produced by every aerobic creature, plant, entity on Earth, anything that consumes oxygen. Because what happens is that when we breathe oxygen and we metabolize oxygen, oxygen is very corrosive. And in the process of metabolizing the oxygen, molecules that are unstable, they're called reactive oxygen species, are formed. And they're kind of like people coming out of a bad relationship. They feel like they've been used and abused, and now they're going to all the wrong places for looking for love. And what they do, it's kind of like going to a bar and hooking up with somebody who's drunk, and then when they leave the bar, bad things happen. Well, when these free radicals are looking for love, they're looking for an extra electron, because the metabolism of the oxygen stripped them of an electron. They're looking for an extra one. So they go and literally hook up with another molecule. They get that electron replaced, and all of a sudden they turn from a relatively innocent substance to something very dangerous, like superoxide. So superoxide dismutase, this job was, as an enzyme, was to fight the bad guy superoxide. Catalase, another enzyme, its job was to fight something called hydrogen peroxide. Now, a lot of women have used hydrogen peroxide for years to dye their hair blonde. Well, what they know now is that these free radicals, this oxidative stress, which is the total damage caused by these free radicals, is the primary constituent in aging. And in fact, hydrogen peroxide free radicals are literally dyeing our hair gray, which is one of the signs of aging as we go forward. And so they knew that it was really important to fight the effect, because even at that point, by the time I met him, they knew that it was not only the primary factor in aging, but it was also tied to over 200 diseases. And the way that they were testing whether their formula had any effect was actually measuring the end result. Because what happens as a result of free radical damage, oxidative stress, as well as something called xenobiotic stress, is that it's basically like these free radicals are hammering on the cells. It's like you're in a castle, you're in a house, and they're just cannons that are bombarding you every minute, every second of every day. And you know, you might have a strong house, and it might the shells might bounce off initially and might not cause much damage, but a repeated pounding will actually cause the walls to break, and it actually causes the cells to die. And you may have heard of something called autophagy. Autophagy is the process the body has of getting rid of old, dead, unused cell parts. And one of the things that they were measuring was the unused or damaged cell parts of our DNA that are measured in what are called T-bars. And so that what they could notice is they said, listen, once you take this product, there's not as much damage to the DNA. The T-bar levels go way down. That's a good thing if your DNA is not being damaged. And so, you know, if it stopped right there, that would have been pretty interesting. But let me give you an example of why I'm glad that they did it. So now we're going to go from Durham, North Carolina. We're going to go from Denver, Colorado, and now we're going to go to Kyoto, Japan. All right. Now this is back in the 80s, late 80s. I'm working in Japan. I'm on the Keihan train line, and I'm passing by a fairly large hill. And at the train station, I look out the window, and there's this beautiful bamboo forest. And going up from the base of the hill towards the top, you can't really see it because it becomes enveloped by the bamboo, are stone steps. I thought, I got to go there sometime. That looks really cool. And so I think it was actually the very next Saturday. My wife happened to be, I was working for a consulting company, and she was back in Utah with our then infant daughter. And so I didn't have anything holding me back. So I went there on Saturday, climbed the hill. It was great. It led to the Iwashimizu Hachimangu Shinto Temple. That was very nice, very at the top of the hill in this bamboo forest. Some of you have heard me tell this story before, but smack dab in the middle of that temple complex was a very interesting monument. And it was a monument to Thomas Edison. And I thought, that's weird. Why is there a monument to Thomas Edison at the top of a mountain in Kyoto, Japan, inside a Shinto complex? Well, let's go back and talk about what Edison's most known for. He's most known for the electric light bulb. You know, he didn't invent it. There had been electric light, arc light, that had been used in Britain since the 1840s, but it was very expensive and it wasn't practical for everyday use. So can you imagine living in the 1800s and everything being very dark and lit by fires and candles and things like that? And he had been experimenting. He decided to concentrate on the filament, what was utilized inside the incandescent light bulb to hold the charge. And he had experimented with a lot of things. And in fact, frankly, he'd gotten up to something where a light bulb could last about 10 hours. Now, a light bulb that only lasts 10 hours, to me, is kind of like giving a guy who doesn't know he's ugly a mirror, unless you can help him. All you've done is now given him a complex. But if you can help him, after now he knows that he's ugly, you can help him say, I can work with that. And you help him get a little bit more handsome, a little bit more in better shape. Now you've done something. And so that's giving somebody a really short-lived light bulb is not very helpful. So he scoured the earth. And what he found in this bamboo forest in Japan was that the type of bamboo in that forest would actually hold a charge and would remain viable in a light bulb for over 1,000 hours, 10 times longer than the earlier light bulb. Now, it really turned out not to be very practical because as excited as the priests were that he found this in their forest, they really weren't too excited about having all the bamboo forest chopped down so that people could have bamboo filaments. And he later found filaments that could go even much longer than that. But aren't we glad? Aren't we glad that he didn't stop? Aren't we glad that we don't have to change our light bulbs every 10 hours? I'm glad we don't have to change our light bulbs every 1,000 hours for that matter. And by that token, I'm really glad that Dr. Joe McCord didn't stop with Protandim, and that he found more and more about it, because science doesn't ever really stop. Discover doesn't ever really stop. It keeps going. And so they found, even during my time at LifeVantage, that the way that it was working was through something called Nrf2. I can tell you, when we first had that conversation in his office in 2008, the topic of Nrf2 didn't come up. He didn't know that. In fact, he specifically told me, we don't know the mechanism of action. Well, Nrf2 has now got a lot of press, a lot of publications. And what's interesting is that he found that not only can we induce Nrf2, which has this cascading effect upon oxidative stress. I mentioned something called xenobiotic stress. That's stress that comes from outside, not even free radicals. That's like if we're eating bad food, breathing bad air, doing things like that. That's an additional type of stress that will also kill our cells and lead to disease. But like that first light bulb, what he found with Protandim is that the effect of the Nrf2 activation in the cells was very short-lived. And so when I met him again in 2016 or 2017, he told me, David, we've got a product, a formula that's 10 times more powerful. I said, well, how can that be? How does that work? And he explained it to me. He said, well, we didn't realize it at the time. In fact, he gave me kind of a backhanded compliment. He used to say, he said, David, you know how I always gave you a hard time about thinking that you were taking too much Protandim? And I said, yeah. He said, well, turns out you were probably closer to being right. He says, of course, I'm sure that was by accident. I said, yes, doctor, I'm sure it was by accident. I wasn't conducting my own individual studies. But anyway, he said that just like the incandescent light bulb filament that only lasted a short period of time, that when they opened the door to the cell so that the Nrf2 could get in, they were also opening an exit. And so almost as soon as it got in, it went out. And so what did they do? Well, just like Thomas Edison scoured the world looking for other things that might be more effective filaments, they looked for ways to block that. And of course, they didn't want to block both things, because then the Nrf2 doesn't get in at all. So they wanted to open the door and close the exit. And that's absolutely what they did. And so by so doing, we now have a product that is really at the cutting edge of research in a whole host of ways. Maybe some of you saw that article that recently appeared in a pharmaceutical newsletter. It was very interesting to me because it talked about the therapeutic effects and potential of Nrf2 activators in respiratory diseases. If you can Google the benefits of Nrf2, like many of you have. And I want to make sure that one thing, we sometimes think that, well, maybe because we're earlier part of LifeVantage, we shouldn't talk about oxidative stress anymore. I'm telling you, yes, you should. And you should try to avoid it every chance you get, because oxidative stress is a big bad guy and we don't want it. And people don't want oxidative stress. They don't want to prematurely age. They don't want the diseases of the body associated with it. And if you have a more powerful weapon in your arsenal to deal with it, you should use it. Now, as I said before, science marches on. In 2008, when I was talking to Dr. McCord, I had a very cool phone. It was a Razor. It had a camera. Wasn't very good. It could text. You could go AAA, BBB, CCC. You remember how we had to text back then? You could do it. You couldn't connect to the internet with it. You couldn't do any type of things. But you could make phone calls. I thought, this is awesome. And then the iPhone came out, and the smartphones came out. Now, as much as I like that Razor, would I go back to using that technology just for old time's sake? No, I wouldn't. How about for loyalty's sake? No, I wouldn't. How about even if it was less expensive? No, I wouldn't. Because I need to have a smartphone. It becomes my office. It does so many other things that I just wasn't able to do before. And that's what I think is really the bottom line with genomics. There's nothing like it out there. There are some things that are similar. And if you want to tell people, if you want to, I'm going to give you a present of my old razor, and I'm going to give you this old technology of Nrf2 activation. That's not a very good present, I don't think, but maybe. And again, I'm not going to say anything bad about it. It's just that science moves on. And what we're finding out is that Nrf2 as the master regulator, not only of antioxidant enzymes, guess what else it does? It also crosstalks to other transcription factors that are related to what it does, but have a slightly different type of effect. So for example, one of the problems that oxidative stress and other types of stress can cause is damage to the mitochondria. That's bad news because the mitochondria is the power plant of the cells, right? And we don't want our mitochondria to go down and be damaged. Well, isn't it amazing that not only can we avoid that from happening by putting up stronger safeguards against the bombardment of the free radicals, but we also have a product in Link that allows the body to create more mitochondria anyway in each of our cells. So it's great how they work together. And there's a lot of cross talking even amongst our products. There's a phrase, so there's 3 words I want you to spend a little bit of time on when you get a chance. One is oxidative stress, which many of you already know about. The other one is xenobiotic. It's not Xena the warrior princess, it's Xeno, X-E-N-O-B-I-O-T-I-C, xenobiotic. That's also a form of stress. Again, it usually comes in the forms of toxins and things that are introduced to our bodies, which can also cause cellular death. And then the other word is cytoprotective. And that basically just means protective of the cells. And at the end of the day, genomics, and especially in combination with all of our other products, is extremely, extremely cytoprotective. Because what it does, it not only strengthens the walls so that when those things are getting bombarded with free radicals like superoxide dismutase and hydrogen peroxide, I said the wrong things too, SOD is the good guy. So when it's being bombarded by superoxide or hydrogen peroxide, it's not only strengthening the walls of the cells themselves, but it's also got its anti-aircraft artillery going, and it's wiping out those guys before they can even make it to the walls. One molecule of catalase can take out 1 million molecules of hydrogen peroxide every second without being consumed in the process. And yet there's still amazingly incorrect information on the internet about antioxidants, even if you look up oxidative stress. And there are what appear to be scholarly articles about the importance of combating oxidative stress. Do you know what suggestions they give you to combat oxidative stress? To try and eat more fruits and vegetables and high antioxidant foods. Going back to that conversation with Dr. McCord, later that night, I had dinner at a kind of a fancy Denver restaurant with Dr. James Crapo, who was on our scientific advisory board. He's a very well-known researcher in his own right. And he said, David, the idea that you can decrease oxidative stress in your body through eating berries, drinking juice, doing anything from outside the body is like taking this glass of water, and he picked up a glass of water from the table, and pouring it into the ocean and thinking you're going to make a difference in the volume that you can notice. He said, you just can't. It's ridiculous. And scientists know it. And yet, isn't it amazing how much misinformation is out there? Well, along those lines, a lot of people for a long time thought that the first superfood, thanks to Popeye, was spinach. Why did they think spinach was so great? Well, it was touted as being a very rich source of iron. Do you know it's not? What happened was that in an early medical study, they transliterated a decimal point about 10 times, 10 places. And so they made this, they said, we got this great discovery about how rich in iron spinach is. It's just not a good source of iron. In fact, the iron that is in spinach is not even very bioavailable or absorbable by the body because of the form of iron it is. Now, don't get me wrong, spinach is good for other reasons, but isn't it amazing about the health myths that persist out there in things that we take for granted each and every day? So as we take down this trip down memory lane, I think it's exciting to know that this research stems back decades. And really, we are involved, we are just 1 or 2 degrees separated from one of the foremost pioneers in free radical biology research. And that we may have benefited from some of his early efforts, but we're now the beneficiary of the latest, greatest, most cutting edge effort to come out of that lab. And I don't know about you, but that's what I want. And I think that's what we should share with everybody else as well. So I think I went a little bit over my 15 minutes, but that's all for tonight. Avoid oxidative stress, stay healthy, and every other kind of stress for that matter, and have a great weekend.